Now, ask yourself this. When your cell culture team needs to evaluate 17 potential quality modulating compounds for a biosimilar development program, how are they doing it? One compound at a time? Or one massive design of experiments with all 17 factors thrown in together? If either of those is the answer, you're leaving months on the table. And you're likely missing the biological interactions that matter most. This episode is about borrowing that same spirit of intelligent parallelization and getting you to your answer in just 2 rounds of experiments. Today, we're talking about one of the most frustrating bottlenecks in biosimilar development: how to screen efficiently a long list of quality modulating compounds, break down under the weight of your problem. You got on one side the one factor at a time, and on the other side a massive DoE, but both will not do the job. Welcome to the Smart Biotech Scientist podcast. I'm your host, David Brühlmann, and this is a 2-part episode. In part 1, I'll walk you through the problem and the conceptual framework we developed to solve this puzzle. In part 2, we go hands-on how to actually build this new framework in your lab. In the 1990s, the pharmaceutical industry went through a revolution in drug discovery. Before that revolution, chemists were synthesizing and testing candidate molecules one at a time. It was rigorous, it was thorough, and it was slow. Then high throughput screening arrived. The idea was simple. Instead of testing one compound at a time, you automate, miniaturize, and parallelize. You screen thousands, sometimes millions of compounds simultaneously. The winning molecules emerged from the data, not from any individual scientist's intuition. That shift compressed decades of chemistry into years. Now, ask yourself this. When your cell culture team needs to evaluate 17 potential quality modulating compounds for a biosimilar development program, how are they doing it? One compound at a time? Or one massive design of experiments with all 17 factors thrown in together? If either of those is the answer, you're leaving months on the table. And you're likely missing the biological interactions that matter most. This episode is about borrowing that same spirit of intelligent parallelization, applying it to cell culture media optimization, and getting you to your answer in just 2 rounds of experiments. Let me set the scene. You're developing a biosimilar monoclonal antibody. Your glycan profile doesn't match the reference product. High mannose is off, galactosylation doesn't align, fucosylation is out of range. These are your critical quality attributes, and the gap needs to close. You do your homework, you review the literature, consult your colleagues, look at what compounds have been shown to modulate glycosylation in CHO cells. At the end of that exercise, you have 17 candidate quality modulating compounds. Not 2, not 5, 17. Now you got option A, which is one variable at a time. Test each compound independently one at a time. This is intuitive and simple to execute. It also takes many experiments, and it will miss every interaction effect. In a system as complex as CHO glycosylation, those interactions are not a secondary concern. They are often the whole story. Option B sounds more rigorous. One large design of experiments with all 17 factors. Statistically designed, combinatorial. In theory, you capture everything at once. In practice, it has 3 fatal flaws. First, you got dilution effects. Adding 17 stock solutions to your medium changes the total volume. That dilutes everything else, your basal medium, glucose, glutamine. Your model is then trying to interpret compound effects against the background that's constantly shifting. The result of that is that your results and your signal degrade. Second, you have to deal with toxicity. Without prior concentration qualification for each of your 17 compounds, some combinations will be toxic. Cells die, wells fail, and the data will disappear. In a clustered, non-random way, with 17 unqualified factors, data loss may jeopardize your ability to draw any conclusions from your experiment. And third, you will have some masking effects. If one dominant modulator is in the mix, for instance a mannose inhibitor pushing high mannose to 90%, that signal drowns out the more subtle effects of the other 16 compounds. The candidates that might give you fine-tuned control will never surface. We ran into all 3 of these problems. And that's what forced us to develop a different approach. The core idea of this new parallel group method approach is the following. Instead of testing all 17 compounds together, split them into 5 parallel experiments, each with 5 factors or fewer. Then, critically run all 5 experiments at the same time. This is not a compromise between one factor at a time and a single large DoE. It is better than both. Here is how we build the groups. The governing principle was the following. Group the components by biological mechanism, not by convenience. Groups 1 and 2 contained the high mannose modulators. Group 1 held the Golgi processing sugars, compounds like raffinose and glucanac that affect mannosylation through osmotic and metabolic mechanisms. Group 2 held the mannosidase inhibitors, compounds that directly block enzymatic trimming of high mannose glycan structures. Group 3 targeted silation and chart variant modulators. Group 4 targeted fucosylation and galactosylation drivers. And finally, Group 5 contain growth promoters, compounds that affect culture performance and modulate glycosylation indirectly through metabolic changes. In every group, we included 2 anchor compounds, manganese and asparagine. These are well-characterized modulators with documented effects across many CHO cell lines. They served as internal calibration references, allowing us to compare results across groups, even though each group was an independent experiment. All 5 experiments ran simultaneously in 96 deep well plates, fed batch cultures, using robotic liquid handling. Why is this design better? Well, on several levels. First of all, on dilution. You're adding at most 5 stock solutions per well instead of 17. The dilution effects are minimal. It's better on masking, because if swinesonin, for instance, is pushing high mannose to 90% in group 2, it only masks the other 4 compounds in group 2. All the other groups are unaffected. It also affects timing and also the time you spend on the experiment, because all 5 groups run in parallel, and so the elapsed time is identical to running a single experiment. You get the biological focus of a small experiment and the candidate breadth of a large one. You run the experiment at the same time in the same calendar window. In other words, the math works out in your favor on every axis. Less time, better signal quality, and better interpretability. Let me talk about another aspect. After a screen like you've just set up, you may be tempted to look at 1 or 2 key glycoforms, for instance, high mannose or G0F. Pick whatever conditions showed the best result and move on. That approach is using about 10% of the information your screen just generated. We measured 13 glycoforms. In biosimilar development, every glycoform is potentially relevant to your CQA specification. So improving 3 glycoforms while worsening 5 others doesn't bring you closer to the reference product. It might push you further away on the attributes you didn't check. To optimize all 13 glycoforms simultaneously toward the reference product profile, you need multivariate statistics. We use 3 tools in sequence. The first is Principal Component Analysis, or PCA. PCA compresses your 13-dimensional Glycan dataset into 2 or 3 dimensions you can visualize. Every experimental condition becomes a point on a score plot. Conditions with similar full Glycan profiles cluster together. Conditions that differ are separated. The key move is this. You project the reference product as an external point onto that same score plot. Now you have a map, and the reference product is the target marked on it. You can see visually which conditions are close to where you need to be and which are not. In our study, 3 principal components captured 76% of the total Glycan variance. Conditions containing 2 F-percetyl fucose clustered far from the reference product target. Conditions containing raffinose clustered closest to it. For the first time, we could see the entire quality landscape in a single picture. The second tool is the Mahalanobis distance. PCA gives you a map, Mahalanobis distance gives you a number. The multivariate distance from each experimental condition to the reference product target. The lower the number, the better the Glycan match. The Mahalanobis distance accounts for the correlation structure between glycoforms, and keep in mind that glycoforms are biologically correlated. If one structural class changes, others tend to move predictably. The Mahalanobis distance treats those correlations correctly, making it a more accurate measure of how close you actually are to your target profile. So you rank all conditions from lowest to the highest distance. The top 20 to 25%, so in other words, the conditions with the lowest Mahalanobis distance become your confirmation candidates. The selection is objective, data-driven, and fully defensible. In our paper, you will find the reference to it in the show note. The conditions with raffinose consistently ranked closest to the reference product, and the conditions with 2 F-percetyl fucose ranked furthest. The third tool is a decision tree. You now know which conditions perform the best. The decision tree tells you why. It takes your Mahalanobis rankings as input and generates a hierarchical set of if-then rules. Which compounds at which concentration level most reliably drives conditions toward the top of the ranking. 2 rules that are non-negotiable. First, always cross-validate. 7-fold cross-validation is standard. You partition your data, train and test iteratively, and ensure your rules hold up on data the model hasn't seen. Second, prune the tree. An unpruned tree overfits your specific dataset and gives you rules that don't generalize. The output is a set of interpretable decision rules you can read out loud, explain to your quality team, and defend to regulatory reviewers. No black box, every branch is understandable and traceable. In our experiment, 3 group winners emerged. Raffinose from group 1, galactose from group 4, enhancer 2 from group 5, combined with a temperature shift to 33 degrees Celsius. We then carried out a shake tube conformation experiment, where 75% of conformation conditions outperformed the best 25% of the initial 96 depot plate screen. So we identified the most optimal compounds and their concentration in just 2 rounds of experiment. Before we wrap up, I want to give you an honest caveat. The statistical tools we used, the PCA, the Mahalanobis distance, the decision trees, were the right tools back in 2017. They still work, but if I were running this study today, I would add one more layer. I would use hybrid modeling. Hybrid modeling combines mechanistic knowledge of your bioprocess with machine learning trained on experimental data. Applied to this workflow, it can do 2 things we didn't do in the paper. First, it can help design a smarter initial 96 depot plate screen by predicting which concentration ranges and combinations are most informative, based on historical bioprocess data you already have. Second, it can minimize the confirmation experiments needed to validate your screen results. I've covered hybrid modeling in depth on this podcast with several guests, including Michael Sokolov, who is also the co-author of this paper. He walked us through the fundamentals in episodes 5 and 6. I also had Chris Gurney, who took it further into the digital twin territory in episodes 173 and 174. Fabian Feidel, also from DataHow, covered the practical side in episodes 99 and 100. And finally, Yossi Quint in episode 137 and 138. All of those are linked in the show notes. In part 2, we go hands-on. Gonna look at how to design your compound groups by biology, how to set concentration ranges without losing data, how to run the 96 depot plate screen with the rigor this method requires. And 3 things I would do differently if I were running this study today. Thank you for tuning in today, and I'll see you in part 2.
All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.